Hello guys, I'm Azim.
Hello, I'm Harry Tonge. I'm from the UK and I studied at Liverpool John Moores University. I graduated in 2019 and I did the international studies at YPC as part of the study abroad program.
Okay, before this, I never even thought that I have UK friends for the first year in Liverpool John Moore. So in 2018, I get to meet Harry during our interchange program.
And that's when I met Azim, and we've stayed in touch ever since. I met Azim obviously 6 years ago, and it's probably a sign that obviously making international friends abroad, but also studying abroad, it's beneficial for everyone involved, but also you make friends for life, and also get to see the amazing Malaysia at the same time. It's the exact same course as what you would study at LGMU. Obviously it's a parallel course, but the luxury is you get to be in Malaysia, you get to meet people for life, and you also get to meet the wonderful lecturers that are obviously here at YPC as well. So yeah, it's definitely recommendable, and obviously with Azim, I've got a friend for life.
Assalamualaikum dan selamat sejahtera kepada pegawai-pegawai PPD, semua pelajar, guru-guru dan ibu bapa yang menghadiri seminar Jom Skor STPM 2024 semester 2 bagi mata pelajaran biologi bersama Mr. Teo. Program ini merupakan program jaringan kolaboratif strategik YPC International College bersama Victoria Institution. Anda bersama dengan saya, Cik Diba, selaku pengerusi majlis pada hari ini. Bagi pelajar yang sedang online, kalau boleh dengar suara saya dengan jelas, minta tolong respon di ruangan chat box. Bersama-sama dengan kita pada hari ini adalah Yang Berusaha, Tuan Abdul Samad bin Uthman, Pengetua Victoria Institution, Puan Salinas binti Saad, Guru Penolong Kanan Tadbir dan Kurikulum Victoria Institution, Puan Zufrizah Aryati binti Muhammad Fazim, Guru Penolong Kanan Tingkatan 6 Victoria Institution, serta guru-guru dari Victoria Institution. Mr. Teo, Guru Mata Pelajaran Biologi, Datuk Dr. Noel Robert, Pengetua YPC International College, Dr. Farah Robert, Pengarah Urusan YPC International College, Kaunselor Akademik YPC. Seterusnya, tetamu jemputan kita pada hari ini iaitu pelajar-pelajar STPM dari seluruh Malaysia. Terima kasih juga kepada ibu bapa dan guru-guru yang turut hadir pada malam ini. Sebelum memulakan agenda utama kita, suka untuk saya ingatkan beberapa perkara terlebih dahulu. Pastikan mikrofon anda dalam mod senyap. Pinkan nama wakil yang akan berbicara sebentar lagi untuk melihat slide pembentangan. Pastikan anda bersedia bersama buku catatan untuk mencatat isi-isi penting. Sebarang pertanyaan boleh terus bertanya di ruangan chat semasa sesi Q&A. Diingatkan kepada semua pelajar untuk tunggu sebentar setelah kelas tamat bagi mengisi kehadiran untuk tujuan sijil penyertaan. Sebelum itu, semua pelajar dan guru-guru dijemput untuk follow dan subscribe media sosial YPC International College di laman sosial Instagram, Facebook, TikTok dan YouTube kami untuk mendapatkan info-info program dan seminar yang akan datang. Sebelum memulakan program kita pada malam ini, saya ingin menjemput dan mempersilakan Puan Zufrizah Aryati binti Muhammad Fazim, Guru Penolong Kanan Tingkatan 6 Victoria Institution untuk memberikan ucapan sepatah dua kata. Dengan segala hormatnya, dipersilakan.
Terima kasih saudari pengerusi majlis, Cik Diba. Beliau juga merupakan pegawai liaison kaunselor akademik yang terlibat bersama dengan segala kolaborasi PTE Victoria Institution dan YPC International College. Pertama sekali, Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Saya ucapkan kepada semua yang hadir dalam seminar online pada malam ini. Saya juga ingin mengambil kesempatan untuk mengucapkan ribuan terima kasih kepada Datuk Dr. Noel Roberts, Pengetua YPC International College, atas kesudian beliau melibatkan PTE Victoria dalam pelbagai program, termasuk pada malam ini iaitu seminar Jom Skor A STPM subjek biologi untuk semester 2, di mana kita akan sama-sama terlibat untuk membantu meningkatkan prestasi subjek biologi untuk semester 2, bukan sahaja untuk murid-murid PTE VI tetapi murid-murid seluruh Malaysia. Kami cukup bersyukur, dan juga untuk segala program yang telah dirancang yang akan dilaksanakan bersama dengan PTE Victoria Institution. Seterusnya, saya ingin memberi sedikit maklumat tentang pengajian tingkatan 6 kepada mungkin hadirin yang tidak terlibat secara langsung dalam pengajian tingkatan 6, dan juga mungkin adik-adik yang baru masuk ke semester 1 yang masih tidak pasti sama ada untuk meneruskan pengajian di tingkatan 6. Jangan pandang rendah pengajian di tingkatan 6. Jangan masuk ke college atau swasta jika dapat tawaran ke tingkatan 6. Kerana lahirnya golongan hebat hari ini sebahagiannya bermula dari tingkatan 6. Sehubungan dengan itu, stigma tingkatan 6 sebagai pilihan susah, pilihan berat atau pilihan akhir harus dibuang jauh-jauh. Ada yang kata buang masa, harus dibuang jauh-jauh. Ini memandangkan sejarah pengajian tingkatan 6 50 tahun telah membuka ruang dan peluang tersendiri untuk lepasan sijil pelajaran Malaysia, SPM, meneruskan pengajian sebelum ke Institut Pengajian Tinggi. Form 6 ataupun pengajian tingkatan 6 ditawarkan di sekolah menengah harian, bantuan penuh kerajaan dan juga persendirian. Hari ini ia dijenamakan semula sebagai pengajian di pusat tingkatan 6 ataupun kolej tingkatan 6. Bukannya lagi di SMK ataupun sekolah kebangsaan, sekolah menengah kebangsaan. Kelebihannya terlalu banyak. Namun terpulang kepada penerimaan seseorang. Nak positif, kita cari positif. Kita cari kekuatan, kita cari kelebihan. Jika terima secara negatif, hanya nampak kelemahan dan kekurangan. Berada di tingkatan 6 sudah jauh berbeza daripada berbanding dengan zaman dulu. Saya sendiri mengikuti pengajian tingkatan 6 pada tahun 1989-1990 dan saya akui ianya agak berat. Terlalu lama tempoh tu, saya rasa saudari pengerusi majlis pun belum lahir lagi masa tu. Banyak perubahan telah berlaku dan pelbagai elemen sepanjang pengajian tingkatan 6 telah diubah untuk kemudahan ramai. Jadi anak-anak, ibu bapa harus tahu pembelajaran di tingkatan 6 atau istilahnya pra-universiti mempunyai kelebihan sendiri. Ia merupakan ruang khusus untuk menyambung pembelajaran ke peringkat yang lebih tinggi. Kos pengajiannya jauh lebih rendah, hanya sekadar membayar sumbangan PIBG berbanding dengan peringkat matrikulasi, praasasi ataupun diploma. Tetapi untuk murid-murid tingkatan 6 selepas mendapat sijil STPM, peluang sangat luas. Bolehlah teruskan ke YPC International College ataupun universiti tempatan, IPTA, IPTS dan juga ke luar negara. Satu-satunya jalan di mana sijil STPM itu akan diiktiraf oleh universiti di UK, di US. Jika adik-adik mengikuti pra-universiti yang lain, laluan pra-universiti yang lain seperti matrikulasi, asasi, itu tidak boleh digunakan untuk keluar negara. Ya. Okay, seperti juga di universiti sekarang, antara transformasinya ialah anak-anak mengikuti pengajaran secara semester, di mana terdapat 3 semester dan setiap semester ada peperiksaan dan setiap peperiksaan boleh diulang atau resit sekiranya subjek atau paper tidak mencapai grade yang dicita ataupun tidak mencapai A. Dia peluang untuk membaiki keputusan tu ada. Selain daripada itu, setiap semester juga ada kerja khusus. Dan kerja khusus ni sama seperti di universiti, di mana kerja khusus membantu menaikkan GPA setiap subjek. Di sekolah juga murid-murid Form 6 diberi kelebihan untuk memakai pakaian casual ataupun smart casual, baju batik, baju tradisional, pakaian selain daripada uniform sekolah, sebagai satu penghormatan kepada kakak-kakak abang paling senior jika berada di sekolah. Di college sudah tentulah. Dan dalam tempoh setahun setengah pengajian, tempoh itu sangat pendek sebenarnya, sebagai satu pengorbanan untuk anak-anak menyediakan diri untuk masuk ke universiti. Paling penting yang harus diketahui, anak-anak dan ibu bapa sekalian, kita ukur baju di badan sendiri. Seandainya kedudukan ekonomi kurang baik, baguslah ke tingkatan 6 dan stay di tingkatan 6. Jika ada yang di semester 1 dan menunggu peluang yang lebih baik, walaupun terima tawaran, saya harap anak-anak stay. Teruskan. Telah terbukti sekarang dengan keputusan STPM yang baru-baru ini kita terima, PTEVI sendiri telah mencatat sejarah dengan CGPA atau PNJK terbaik yang pernah dicapai, iaitu 3.12. Keputusan STPM kebangsaan juga telah kita atasi nasional, iaitu pada 2.84. Sebanyak 1,116 calon ataupun 2.7% calon mendapat PNJK 4.0. Apa maknanya? Tepuk dada, tanya selera. Jadi ayuh anak-anak semua, kita buktikan kita mampu berjaya menerusi pengajian di tingkatan 6 dan STPM. Sekarang juga terdapat banyak bantuan dari segi kewangan jika diperlukan. Okay, dengan itu, saya tamatkan ucapan saya pada malam ini dan saya ucapkan semoga usaha anak-anak selama 6 bulan, 5 bulan, 4 bulan ini akan membawa kejayaan yang cemerlang untuk semester 2, khususnya kepada anak-anak yang mengambil subjek biologi. Okay, so good luck to all of you. I believe in you, especially Victorians, and all of you taking biology semester 2. Enjoy the session tonight. You're very lucky to have Mr. Teo, the best teacher, bio teacher in KL, to share his experience and his knowledge with you. Don't let it go to waste. And after the session, take a good rest, okay? Do not stay up all night studying. You've studied enough. And for tomorrow, make sure you pack everything tonight. Have a light breakfast. Leave the house early so you're not rushing. Don't give yourself added stress or pressure. And I'm sure all of you will do excellently. That's all from me. Assalamualaikum. Thank you very much.
Terima kasih diucapkan kepada Puan Sufriza atas ucapan yang sangat inspirational sebentar tadi. Seterusnya, saya ingin menjemput Miss Aminah, pensyarah daripada YPC International College. Dipersilakan.
Okay. Bismillahirrahmanirrahim. Assalamualaikum dan salam sejahtera kepada semua pengetua dan guru-guru, Datuk Dr. Noel Robert, Dr. Farah, YPC staff dan para ibu bapa dan juga adik-adik yang join program kita pada malam ini. Hopefully semua dapat dengar suara saya dengan jelas. Okey. Pertama sekali, izinkan saya memperkenalkan diri. Nama saya Puan Aminah. Saya merupakan salah seorang lecturer dan juga program koordinator di YPC International College. So, pada malam ini, saya ingin berkongsi sedikit tentang one of our most popular and in-demand program offered by YPC. Program yang akan saya kongsikan pada malam ini merupakan degree program from Liverpool Jom Skor University UK, iaitu Bachelor in Science with Honours in E-Business Technology and Management, atau singkatannya kami panggil BEM lah. Okay, for your information adik-adik semua, this degree is a multidiscipline. Maksudnya dia combine dua area of study. So untuk BEM, it cover IT and business. market. So if adik-adik minat bisnes but on the same time adik-adik nak dapatkan kelebihan dalam bidang IT, BEM adalah pilihan yang paling tepat lah untuk adik-adik semua. Sebab apa? Sebab melalui BEM, adik-adik dapat mempelajari front-end aspect of e-business seperti digital marketing and sales, as well as back-end IT aspect seperti web development and database management. Because melalui program ini, adik-adik dapat belajar subjek seperti e-commerce, website design, database design, system design dan juga software development. Dan untuk maklumat adik-adik, this skill are very essential for anyone yang ingin berkecimpung dalam bidang e-business industry, whether as a business owner ataupun as a professional in a technology company. Selain itu juga, our certificate is a UK degree certificate. Maksudnya our degree certificate bukan hanya diterima di dalam negara Malaysia, tapi turut diterima di peringkat antarabangsa. Dan untuk adik-adik, contohnya macam yang ada cita-cita nak bekerja kat luar negara ke, sambung belajar kat luar negara ke, ini adalah peluang untuk adik-adik mencapai cita-cita adik-adik semua. Okey, di sini izinkan saya berkongsi sedikit tentang our alumni and what career they have secured as a BEM student. Okay, next slide please. Okay, ini contoh our alumni dalam bidang BEM, which is kalau adik-adik tengok kat sini, Gitavi, she is a principal consultant at Manhattan Associate Singapore. So sekarang dia bekerja kat Singapura lah. Sama juga dengan Wayne Lim, which is he is a senior account executive at MAT Thailand. And Suk Basing, he is a global payment manager at Z Group. And for Navin, he is an international sales manager Amazing Fruit trading at Dubai, UAE. Kerja dekat Dubai. Untuk Gajendra Gurunathan pula, he is a senior IT analyst at Deloitte. Deloitte ni is a multinational company yang based dekat UK. Okay. Untuk Intan Baiduri Kamalu Bahari, she is a senior system support at Berjaya Sompo Insurance Berhad. So dari sini adik-adik boleh tengoklah contoh apa career yang they had secure as a BEM student. Go for the next slide. Okay, this is our alumni yang graduate on July 2023. Okay, kalau adik-adik tengok kat sini, kumpulan ni terbahagi kepada dua. Ho So Chet and Tan Yuan Yi, they are more towards IT. Kalau adik-adik tengok, Ho So Chet is a software engineer at Dina Front System Sendirian Berhad, while Tan Yuan Yi is a front-end developer at TDC at Malaysia. And for Zixin, Ko Hua Han, Ellie and Sim Mei Teng, they are more towards business, which is for Zixin is a service marketing executive at Hatseng Star Sendirian Berhad, company besar juga lah. So Kon Hua Han is a QA/QC specialist at Flash Express Malaysia, and Ellie Lim Yi Teng is a procurement at Genting Malaysia Sendirian Berhad, and Sim Mei Teng is a HR executive at Vertex Mastering. Okay, so if adik-adik berminat dengan program yang kami tawarkan ini, especially lah untuk lepasan STPM, ini adalah kiranya shortcut untuk adik-adik. After habis STPM, adik-adik boleh terus sambung ke peringkat degree level selama 3 tahun sahaja. Dan jika adik-adik nak tahu info dengan lebih lanjut, adik-adik boleh contact our counselor dekat ruangan chat yang disediakan. So saya rasa that's all from me. And berbalik kepada program kita pada malam ini, saya ingin menjemput guru yang akan mengajar pada malam ini, iaitu Cikgu Teo yang merupakan guru daripada Victoria Institution. Beliau sudah berpengalaman selama 11 tahun dalam mengajar. Jadi saya menjemput Cikgu Teo. Silakan Cikgu.
Okey, terima kasih. Semua boleh dengar kan?
Boleh Cikgu.
Okay. So I believe you all can see the screen, right? Hello?
Yes, can.
Thank you.
Okay, so I will start now because no worry, you can call me Mr. Teo. So I know tomorrow is the exam, but please keep calm. Usually from what the analysis I observed, the questions they ask of late, they are all simple questions. But you please be reminded that quality answers matter. So important thing is when you receive the paper tomorrow, just read everything, you spend 5 minutes. Then for the 15 objective questions, you have to really pace it up and do it within 20 minutes. Then for the structure and essay, each one allocate around 15 minutes. So you have, because there's 2 essays you need to answer, actually there are, if there's 2, that means it's 4. So around 15 minutes each essay. So general rule, I will go through general rule first. Do not give, let's say they ask you for structure. For example, they ask for, give the 2 hormones, name the 2 hormones. So you write there estrogen, progesterone. Remember, this is still a structure question. So do not go and answer until half a page. So important thing in Form 6, especially bio, actually to score, it is not easy because you need to know all the terms, the rules of the terms. Like for example, posterior pituitary gland releases. Please take note. It is not secreted, it is releases. Only 2 types of hormones which are released by posterior pituitary gland, which is oxytocin and antidiuretic hormone, ADH. So please take note of the word releases. Then for the insect, the neurosecretory cell, it is produces. Later, I will show you the MPM report, which they believe that the terms used must be correctly used. So general rule, scientific terms must be spelled correctly. For example, voltage-gated sodium ion channel. The word ion is very important. And this is the way we write for this is the way to write your IgM, IgA, IgG. Then for interleukin 1 and interleukin 2, please take note how to write it. Then antigen presenting cell, APC. So for clonal expansion, in Campbell, they use clonal selection and other book use clonal expansion. So, but certain years, they prefer, because clonal selection, clonal expansion is a general name. Some years, they prefer it to be, because clonal means making clones. But the word, that's why I always ask my student, please use the word clonal expansion, then in bracket, you write proliferation and differentiation, because you know your helper T cells, they undergo clonal expansion. That means they proliferate to produce more, then later they differentiate to become effector helper T cell. The word I will prefer my students to use will be effector helper T cells and memory helper T cells. So usually I will ask my students to write this term all together, because Campbell write clonal selection and other books write clonal expansion. So as long as the word proliferate and differentiate is there, that is the keyword. You need to use the terms correctly. So effector T cell, effector B cell, B lymphocyte, T lymphocyte. Then for scientific name, you cannot write italic. So please underline separately. That means you know I think you will learn this in your Form 4, Form 5 already. So general rules, I'm skipping this one. I have no time for all this one. This I think basically in Sem 1, you already listened to all these things. Crossing out, no liquid paper, nothing. Okay, one more thing you have to understand. The bio paper is marked online. So your paper will be scanned. So please be careful. A lot of you see, if you write your handwriting, please take care of your handwriting. But I know when there's no time, you write fast. So please be careful. Your paper will be scanned. So let's move on to section B, the structure question. If the question asks you the antibody, the name, if they ask you to give the name, you have to spell the thing out. You cannot write, like for instance, this one is immunoglobulin G. You cannot write this one. If the question say name, you have to write the name. So what is meant? If the questions ask, the next type of question that they ask in structure is what is meant. That means definition. So you always ask, what is meant by immunity? Define immunity. Immunity is. You always write complete sentences. Please do remember, you need to write complete sentences. So the next question, state, describe. This one I will tell you in a bit while. For example, this question. Please take note. Oxygen dissociation curve in chapter 7. This one came out already. This one came out and the report came out in 2019. The STPM 2019, the report already out. So when the question asks for three factor, please take note. Three factor. A lot of students, they go and write the three factor. pH, partial pressure of CO2, temperature. This one, you will not get the marks. Candidates only mention the factors, but the acceptable answers were, you have to have like the kata kerja. It's like low pH, high partial pressure of CO2, PCO2 high. Is it high or low? So when the question asks about factor, you have to state the factor. You have to write a statement in one sentence. Then for describe, I think you already know, explain. So please take note. I'm not going to cover this later already. Oxygen dissociation curve. This one, the one came out, it's already come out in structure in 2019. And the one came out in essay is comparing between fetal hemoglobin and maternal hemoglobin. So I will assume that all students have to know how to answer for myoglobin. Myoglobin. It is important for you all to know the full essay on myoglobin. You have to understand, for hemoglobin, there is molecule cooperation. But for this one, because our myoglobin is only accepting one molecule of oxygen, they will be fully saturated. So please take note. If comparing hemoglobin, comparing the curve, you must mention the ODC curve for myoglobin shift to the left of the ODC curve of hemoglobin. So you have to use the correct term. You cannot say myoglobin shift to the left. What does that mean? Please be careful when you write, because quality answers matters. So section B for structured question also have explain and differentiate. And if it's differentiate, you have to write, use the keyword. I will always ask my students to use the keyword but, while, whereas, comparing the same thing. So for example, this is the question that came out already for Evelus. This is chapter 7 again. So what is the rule? I know. Actually, don't be worried for tomorrow. Of late, the question comparing, I make analysis before the new syllabus. The new syllabus that they came out since the 2012 one, look carefully. Every question, it is actually very simple. It is actually very simple. So the one that differentiate you between good students and better students, it will be you cannot know what the marking scheme wants. Sometimes we think we write enough, but like for example, this question, B2. They wanted prevent the collapse of alveolus fall due to high surface tension of water at the end of expiration. So usually students will write collapse of the alveolus wall at the end of expiration. Yeah, but that year they prefer a complete one. Sometimes the answers are very complete. They demand a lot. But never mind, there is only 1 mark. Let it be. Move on to the next question. So for essay, this one state is the same reason. So if they ask you to define, let's say the homeostasis. Homeostasis, importance of homeostasis came out last semester. So for this part, if they ask you to define homeostasis, so you have to take from Campbell, the 12th edition. Campbell, have to use Campbell. It is the steady, you see, the steady state of physiological condition of the body refers to the maintenance of internal balance in achieving homeostasis. Animal maintains a relatively constant internal environment. So actually, you have to have in learning bio, later for Sem 3, please have a mental image. So when they say homeostasis, you have to have a mental image of the page in, if you can remember the slides from your teachers. So steady state physiological condition to maintain internal balance by having constant internal environment even when the external environment changes significantly. So you can use your own word, but the keywords are all there. The keywords are all in the blue. So describe or explain with the aid of label diagram. This question is very important. If they never say with the aid of label diagram, don't bother to draw. There's no marks given. Don't waste your time. They don't mark the diagram at all because they say, if the question never asks with an aid of label diagram, you go and write everything. Let's say, for example, Cori cycle, ornithine cycle, but they never ask for it. They still want to see your essay. So this one, distinguish, differentiate, compare. Please give complete sentences. Calculate. So usually for calculation in Sem 2, there are how many calculations altogether? So for example, calculation will be lung volume capacities and capillaries, calculating your net filtration and growth curve. So in Sem 2, there are only around 3 main calculations for Sem 2. So let's move to chapter 7. Okay, chapter 7, the first part, transportation of carbon dioxide. Before that, are you still with me? Okay, look at this analysis. Look at this analysis. Let's start with chapter 7. Okay, chapter 7, that's why I say alveolus. So if the question on alveolus came out in chapter 7, it will be most likely an essay on the structure of human respiratory system, asking what is the function of surfactant and so on. Then the one you have to pay attention is the structure of hemoglobin. Then if they ask you, they can relate to the oxygen dissociation curve of myoglobin, because it is very unlikely for fetal hemoglobin and maternal to come out again, because can y'all see the screen? Can y'all see this screen? Okay, I believe y'all can see. So for this part, if you look carefully, oxygen dissociation curve, the one that came out is fetal. So please take extra note on the structure of hemoglobin. This part is still empty. And also this part on myoglobin. So transport of carbon dioxide, I will go quickly, but it is nevertheless, I always say, study all. I never spot for my students because it is very difficult to spot. Breathing cycle, look carefully, the one in essay, breathing cycle. Later I will mention, if you did not memorize the definition based on the book, you cannot do that question. If you just write the formula, you will not gain the marks. Then structure for stomata. Stomata opening, 20 ulangan came out. So I believe is the stomata closing during water stress or high transpiration. So this is the analysis for chapter 7. So now we go to the PowerPoint. Now again, we go to the PowerPoint. The first part, how carbon dioxide is transported. So I will not pay so much important, but in the picture, you must take note. When you explain, you have to know that this is step number 1. Then you have to explain this is the first part. Your CO2 plus H2O, carbonic anhydrase enzyme, hydrogen carbonate, come out, H plus. This is the second part. Please take note, second part. When you see this picture, if this come out in structure, it's either this picture or this picture. There are only 2 pictures that come out for transport of carbon dioxide. So for this one, you have to know which is the first action. This is first in the respiring tissue, CO2 comes out first. Then oxygen goes out, oxygen. Then in the alveolus, O2 comes into the your red blood cell first, then later CO2. Look at this one. Please do not confuse when you study. So basically, this is extra for those who want to score. So you read what is Bohr. So if they ask me, explain how the partial pressure of carbon dioxide affects the control of breathing mechanism, it just recently come out. So you can later refer to this one. And to me, this is the additional stuff. So now back to this question. If you cannot memorize lung volume, there are altogether 4 lung volumes and 4 lung capacity. Most likely it will be 8 marks. But if you cannot memorize the definition, please do not attempt to do this question, because I believe you will get no marks there. So this one already come out in structure, and this is the formula. This is so-called the formula that you all use when calculating. So I believe if this question come out in essay, writing the formula will not get you the marks. So please be careful. I believe all your teachers already do all the past year with this one with you already. So and the last part for chapter 7 is the closing stomata. Opening of stomata just recently came out. So please take note. Closing of stomata. Closing of stomata, they will ask you, when does it happen? Especially if the question asks during high transpiration or water stress. So you must mention, the first thing you must mention is the ABA. So what does ABA stands for? You must know. Then who produced these ABA hormones? So from here, this one, it binds to the ABA receptor at the gut cell. So what does that cause? So they cause the influx of calcium. Actually, the vacuole also releases calcium. Then this influx of calcium causes chloride to come out, and later through facilitated diffusion, K plus comes out. So you have to write all these things. So please take note. Proton pump. You have to know proton pump is inhibited. Inhibited. Please make sure you know when calcium goes out, calcium comes in, your proton pump is inhibited. So that is the keyword. So I'll move on to chapter 8 because now is 8:42. Chapter 8. For chapter 8, okay, for chapter 8, cardiac cycle, please take note. Cardiac cycle in Campbell is quite limited, but you please take note that Campbell is only one of the books. So if the question asks on cardiac cycle, but before that, let us refer to this part first. Look at this one. Describe the structure of mammalian. This one came out last semester. So the most likely of this question will come out, it will be this part, initiation and the whole cardiac cycle, ventricular systole came out, ventricular systole. So I use a red one, ventricular systole in 23 ulangan. So please take note, the initiation and regulation of heartbeat. That means the intrinsic, like whatever the Sasabadi book call, intrinsic and extrinsic. So this one, please take note about that one. Then hypertension, the cardiovascular diseases. So for cardiovascular diseases, you just need to read all. Then lymphatic system in relation to blood circulatory. This one is regarding immunity. So this one regarding blood circulatory system. So, and the last part, the movement. This is 2013. So for this part, 8.1, for we won't go to the plants yet. Let's go on. Okay, for this part, cardiac cycle, you have to know it consists of three phases. Your atrial systole, ventricular diastole, atrial and ventricular diastole. The third one, please take note of these three. Then you have to explain. Then you ask me, how much detail you need? It depending on the marks, how much detail that you need. So in Sasbadi, they give this thing as well. Isovolumetric contraction, ventricular ejection. So that year, they asked about ventricular, this is your ventricular systole. So if the question asks about ventricular systole and is 4 to 8 marks, so I believe you have to write in detail. So I will pay more attention to the regulations. So you have to know there are two parts. Intrinsic control is your sinoatrial AVN, then Purkinje bundle his, then both ventricles contract, apex up. This is the thing. Then extrinsic control, there are two. One is to increase, the other one is to decrease. The main purpose to increase your heartbeat, the other one is to decrease. So where is it? Everything is in detail from stretch receptor. This is your cardiac accelerator. Then your center. Then through sympathetic nerve. So there is this is nervous control. The other one is your cardiac. This is your nervous control. Then this is the hormonal control. So basically, I believe all of you have already know this because it's quite detailed in your Sasbadi book. So I'm quite okay with it. Usually, I never ask my student to refer to Sasapadi book. I ask them to refer to Campbell. Okay, the cardiovascular diseases. So every year, the reports comes out and I make analysis of the report. So please take note. You see, student lost a lot of marks. Actually, they lost marks to incomplete answering, hardening of artery, and loose elasticity. So they must know what is arteriosclerosis and what is arteriosclerosis. So both arterial is the most common type of arterial. So this is arterial first, then later under it you will have arterial. Arterial is about the plaque formation. So R come before T. So arteriosclerosis is the general one. So please take note. A lot of students right there, narrowing of blood capillary actually is narrowing of the arterial lumen. Then what does that cause? They cause students will write block the blood flow. Actually, it did not block. If it's blocked, you already die. It is actually restricted. It became narrow. It became restricted. So plaque formation is formed in the inner arterial wall. So please take note. Then these are the same thing. These are the same thing. Plaque build up in the coronary artery, restricted blood flow. So don't use the word block. Restricted blood flow. Or you can use the word obstruct. But certain year, you see, when students, I believe there is a chat. Oh, sir, can we write tumica intima instead of inner wall of artery? Yes. But please make sure you don't write it wrong. So obviously you can. So we don't know. You see, look at this part. Obstruct the artery. But for this heart myocardial infarction, they want you to know the coronary artery. So sometimes the things are very, very detailed. So they want all these things. It's in the reports already. I believe you already know all these things. So today, just relax and take this lesson as a revision. If you don't know anything, there's still time for tomorrow morning. The exam is afternoon, tomorrow afternoon. So you are still having ample of time. So look carefully for this part. Arteriosclerosis is the thickening, hardening, and loss of elasticity. So you see, look at I write number 1, number 2. Then it is a condition that causes the lumen of the arteries to become narrow. Narrowing of the arterial lumen, narrowing. Then restricting blood flow. And if you want to talk about arteriosclerosis is the plaque formation. So for causes, please, if in exam, they ask you what are the causes, and if you write one word answer, causes, unhealthy diet, comma, all these things, try to, if the question asks you to describe or explain, describe is you write one sentence. Explain, you write multiple sentences. But try not to give a one-word answer. Comma, you think you will get 5 marks for that? So try not to give one-word answer. So you see, for example, you know that you are writing all these things. Methods of prevention. So a lot of students believe they write a lot, but they fail to classify it into a group. So because maybe they want, maybe it depends on marking scheme. maybe they want healthy diet, only 2 or 3 explanation. Physical exercise, they want 2 or 3. And healthy lifestyle, 2 or 3. Or they want each one 1. Each one 1. So it depends. So when you read, you have to know it's the same like chapter 13. Prevention of infectious disease. The social, economical, and biological. So you have to know how to group it into, because we might not know. When we answer, also we need to group it to show that we are STPM student, bio student. So the next part is on this question. This is the blood capillaries and tissue fluid. So you have to understand the concept first before you attend. This one came out in objective already, and there are many came out in the previous objective. So I have no worry. You only need to memorize the formula: HP blood minus HP interstitial, OP blood minus OP interstitial. I believe you can do this question. Use the formula. Always use the formula. Try to use the formula first, because I believe you can get the answer. If not, then you try to understand the thing. This is blood flow. You know blood flow is from arterial to venal. So first thing in the question, usually in the question like this one, all came out before already. So like for this one, you have to know the one having high hydrostatic pressure, high HP, will be the arterial end, and venal usually will be the low HP for this side. Now, the next question, what is hydrostatic pressure? That means it's the force that forcing out. OP blood is the resistant. Then for HP interstitial is force out into the blood from the interstitial, and OP interstitial is the resistant. So always use the formula. Try to use the formula. If not, then you see this question came out in, this is from one of the book, I think it's from Oxford, this one. So you can get the answer. This is the student's book, so I snapped from it. Because I want to tell you that, you see, from this side, you will know this is the arterial end because this HP is high. The HP here is low, this is your venal end. So you use the formula, then you can calculate. For this part, it is also the same. Now, this is the 2021 question 5. So which one is true about X and Y? From there, you already eliminate 2 answers. Here, you can know that from here, look at this part. Here is 35, here is 15. So Y will be venal, confirm. So it's either this one or this one. So net absorption, net filtration. So use the formula. If you cannot use the formula, look at how I do it. So you can use your logic, push in, push out, up to you, whatever it is. So I'm going to skip this, all these things, because I believe you already do it. So now I will move to this part where for the plants. It is an important thing that you know for the plants as well. 8.2 is a transport system in vascular plants. So the first one is you must know what is the root hair, then the three theories: root pressure, cohesion tension theory, and transpiration pool. Look at this part. Simplest, the three movement, you must already know apoplast, simplast, and vacuolar pathway, the definition by heart, came out in structure, came out in essay recently, so shouldn't be any problem. Then plant roots, mineral, water dissolve, this one in essay. Please be careful, this one, essay. So for this part, the three theories, if you're looking at these theories, transpiration pool came out, cohesion, tension, transpiration pool, same again, cohesion, tension, then root pressure and transpiration pool. So basically, all these things came out already, but this is the big question for your structure. So for this part, mass flow, the next part is, this is transport of water. Now we move to transport of food, the translocation of sugar. So 2008 is the one. Later, we'll see. Mass flow, you have to master. The next will be your electro-osmosis. Cytoplasmic streaming, describe. So they are altogether 4. 2008 is a long time, and this one still haven't come out. The concept of mass flow, Munch model all came out. Munch model is quite old already, so please pay attention. Then translocation, just recently also. So you have to know which one is a soft cell, which one is a sink, potato tube germinator, which one is a source, which one is a sink. You have to take note. So based on this one, let's say go to the first part. The first part will be this one. Campbell, cation exchange. You see, what happened in the root hair? The roots acidify the soil solution by releasing CO2. So the H+, look at the H+. The H+ here, the H+ is going to release, then they neutralize the charges on the soil particle, releasing, for example, this one, calcium, to be reuptake into the roots. So please take note, active uptake of mineral ions. Please take note. So this one, look carefully, the proton pump. So conclusion, I will go to conclusion transport of mineral ions. Let's say this is your root, this is the endodermis, this is the xylem. Please take note, active transport. Active transport, the first you have to explain about carbonic acid exchange theory. Then this is the first part. It's happened here. Happened here. Number 2. Look at number 2. Number 2 happens here. So cation exchange in soil, that is number 1 in carbonic acid exchange theory. Please take note that it is in Campbell. So have to read. Then the second thing, actively loading mineral salt into root xylem. Please take note. You have to be precise. At this part, there is root xylem, stem xylem. Root xylem, stem xylem. Then for this part, facilitated diffusion. This is at the cortex and pericycle. This is area number 3, area number 4. So area number 3, area number 4. So you need to know, after the mineral ions diffuse into the root, the ions diffuse down a concentration gradient. Okay. So whatever pathway they choose, you have to explain. So this is about transpiration pool. So a lot of people, they are asking, are we supposed to read everything in detail? Yes, because it is in Campbell. So in transpiration, water vapor, you have to know water vapor. This is transpiration pool. The whole essay on transpiration pool, 6-7 marks, came from here. So you have to know water vapor. Then there is, look carefully, water vapor diffuses from the oil air spaces to the leaf to the drier air outside. Then is replaced by evaporation from the water film that coats the mesophyll cell. Then it causes the air-water interface to retreat further into the cell wall. The curvature increase the surface tension. Then they start pull water surrounding cell and air spaces, and eventually they pull from the leaf xylem. So please take note, this is leaf xylem. Now, this is the stomata closing and opening nothing. Now, look at this one. This is the cohesion tension theory. So, cohesion tension theory, although they ask for cohesion tension theory, most of the students will write number 3, number 4, number 5, number 6. They actually fail to tell 2 more things. Number 1, where is the pressure come from? Where is the negative pressure come from? You have to say about a bit about the transpiration pool. At the evaporation, lower the water at the air-water interface. At a negative pressure that pulls throughout the xylem, through the transpiration pool, the theory first part. Then also talking about bulk flow. So these are the one that you have to take note. But this one recently came out already. So if I want to know the one that we have to be careful is this picture that can come out in structure, and also this one can come out in structure if it do come out. So the next part, translocation. Translocation, you have to know the mass flow hypothesis. So what is companion cell, modified companion cell? You have to know. Actually, there is 3 parts. Once you understand all these 3 parts, you have no problem. So the first part is here. At the source, what happened? Sucrose is actively loaded from the source cell to the companion cell to the, this is the sieve tube. When they are thus, when they have a lot of sucrose there, the solute potential increases, thus lowering the water potential. So what happened is water from the neighboring xylem vessel will diffuse into the sieve tube by osmosis. So you have to remember all these things. It is like this is the first part. Then here, after you talk this first part finish at the source, then you talk at the sink. Because when you write at the source, the ending paragraph will be, it creates a high hydrostatic pressure. Then at the sink, sucrose is unloaded to the sink cell. So when this unloaded, the water potential higher, water goes out. This creates a low hydrostatic pressure. So the second part, the second paragraph, you talk about a lower the hydrostatic pressure. then there is a potential, the third part, there is a potential difference. So when there is a potential difference, things will start to flow from a high to low. So you need to know all these things. So this is known as the pressure flow. So Munch model, please read on your own. Munch model. It is almost the same thing, but this is experiment in the lab. Experiment in the lab. So for the one I want you all to pay attention, maybe is the electro-osmosis. It's still in your syllabus, but obviously it's not in Campbell. So this part, we will refer to this one. This part, it came out, this is the one from the CD, from the ministry. So step number one, you see, K plus is actively loaded into the sieve tube. Then they accumulate near the sieve plate, drawing water and sucrose. When they go down, look at the animation. Now I click step number 2. They will rush down because there is a potential difference. Then later, the step 3, reuptake of the potassium ion. So this is the one. So let me go back to the slide. So the first thing is potassium ion is actively loaded. This is number 1 from the companion self into the sieve tube. This increases the K plus, the positive charge at the, you see, at the sieve tube. On top, there's a positive charge, and below it become negative. So there is a potential difference, and it causes the ion to charge through. So that's why they are known as electro-osmosis. So you can read more, I think is there in your Sasbadi book as well. So for chapter 9, this is chapter 9. Chapter 9, nervous system, until now haven't come out. So if you have the time, then you read. So resting potential, rising and falling phase just recently came out. Then action potential across the axon. Explain how resting potential is maintained. The speed. Describe the structure of synapse and neurotransmitter. Okay, this one, essay. So along the axon, and please, non-myelinated came out, synapse came out, synapse again, myelinated, but it's 2007. So please take note on the essay on transmission of impulse along the axon. Is either along the axon is either myelinated or non-myelinated. Across the synapse 2012, so it's time. So if this question do come out, please take note. But because 2015 came out the neuromuscular junction, so 2019, so we see there's so many questions that haven't come out. So muscle contraction, sliding filament theory. So this one we will still, that's why I believe you have read everything. So don't be worried. This one, if you don't read on sympathetic and parasympathetic nervous system, you cannot answer this type of question if you don't read. Then curare and oxytocin, this one I believe all almost came out already, but still I will go into chapter 9. Okay, the first part of this one. Students always talk about this part. This is your resting potential. Resting potential, that means your sodium potassium pump. I believe this part you already master it very well. Please take note how to your 3 sodium ion out, 3 sodium ion out, 2 sodium ion in. This is the first factor that creates why inside is negative. Why inside is negative. The other reason, there are other reasons as well. The leakage. The leakage is the second one. Leak channel, leak channel. Please read more on the second one. And the third one is they got abundance of negatively charged protein. That's why it contributes to the negative charge inside. So if you cannot, just remember, you already are able to answer already. That is additional things. So if you cannot, if you already master this side, I say you are very good already. So please take note between if they're asking you for phases or process. Resting stage. The other one, process. Process will be depolarization, repolarization, hyperpolarization. So the next part, this is the, actually, this will be the notes on how to write for essay for transmission along an axon. The other one on the left is non-myelinated, on the right is myelinated. So transmission of impulse at the synapse. So look carefully. When failing to write this one, action potential arrive at the axon terminal or your presynaptic node. It depolarizes the membrane. Please take note, it depolarizes. When impulse arrives at the presynaptic node, it depolarizes the membrane. It causes your voltage-gated calcium ion channel to open. It causes the influx of calcium ion. What do calcium ion do? Directs or signal the synaptic vesicle containing neurotransmitter to fuse with the presynaptic membrane. The vesicle fusing to the, thus releasing the neurotransmitter into the synaptic cleft. So this one, voltage-gated calcium ion channel, voltage-gated sodium ion channel. Please take note of the names. The one here is ligand-gated sodium ion channel, because the one binding is the neurotransmitter chemical, ligand-gated. So please take note of all these things. Muscle contraction. This is the the essay on muscle contraction. So students will miss out a lot of things, especially when they write muscle contraction. So please write from neuromuscular junction. No need to write the whole thing. No need to write the whole thing. From the motor end plate, you can start from the motor end plate to the sarcolemma. No need to write the whole thing from the presynaptic node. That is your neuromuscular junction. Now, if the question asks you for muscle contraction, you answer about muscle contraction only. So this is the structure question that I come out from my school. I think most of the students cannot draw, but I don't think so. They will ask you. So if you are able to know how to draw the length and everything, that means you understand what is Z line, A line, I line, H zone, and your M line. So please take note of this part. Yeah. So when they ask for muscle contraction, you can talk about these parts. This part. So neuromuscular junction, you talk about this part. The motor end plate one. So please take note of muscle contraction. For muscle contraction, look at this one. So myosin head attached to the actin myofilament. So myosin head attached to the actin, forming this one. It is very clearly said for muscle contraction. So ATP binds. All these things, I think I don't have time to continue with such detail. So I believe your teacher already guide you in this one. So I believe I will skip. So usually this part will be around 4 steps. And please memorize the 4 steps. Then the next one. For the sympathetic and parasympathetic nervous system, a lot of students, they write this answer. If I come out in a test or whatever, they write the effect. So effect, yes, effect, you might have only 1 or 2 marks, but you will not get a lot of marks. Effect will only give you a few marks only. Because if they ask you to compare, this is the comparison. Origin of neuron, position of ganglion, length of fiber, neurotransmitter, condition when active. This is the, most of the students only write this one. General effect. And all these things. So that I believe the marking scheme, there will be a limited, that means they will give students can have a maximum of 3 effects only. So if you don't memorize this one, or you don't know how to compare between sympathetic and parasympathetic nervous system, if you don't read, you cannot do this type of question. Always remember, of late, I can tell you, later tomorrow you will see whether I'm telling the truth or not. They ask simple question only. Now, next one, dopamine and curare. So please take note of all the keywords. So what happened here? So you see, cocaine. This is your dopamine, the neurotransmitter. So when this is transmitter join here, so when the transmitter dopamine, so it will polarize the membrane, giving rise to the impulse. Now, dopamine blocks the reuptake. What does that mean? It blocks the reuptake. That means there will be continuous stimulation. So the continuous stimulation will give you pleasure, will give you euphoria, state of arousal. Okay, so the pleasure is here. Then the accumulation of dopamine will give you the heightened pleasure. Oh, you love it, the heightened pleasure. So what happened is, when suddenly your body, how do I put it? This constant pressure, your body will respond to the drug. What do? They produce less receptor. So what you do, you take more cocaine so that you want to get back the same amount of pleasure that you have last time. So then you have to talk about addiction. Then later, if the removal, sudden removal, you talk about withdrawal. So this is the effect of cocaine. So mechanism of action, this is the mechanism of action, then the effect. So you have to explain in detail if this question come out. So the next one, steroid hormone and non-steroid hormone. If they ask me for steroid hormone, okay, this is the answer. 1 to 5. Everything is there. Steroid hormone action. So I believe if they want the question example, use testosterone, because testosterone is muscle protein. So it's a very easy example you can use. Then for this one, non-steroid, you use another example, which is your adrenaline. It's so easy, adrenaline. So you have to understand how is the process. This is also, I think this is in Campbell. So what happened is, this is your first messenger. It binds to your GPCR, forming, you see. You can write there, hormone receptor complex. It's true also. So when the messenger binds, it activates your GPCR. So it binds to G protein. Then the next one, GTP binds to the G protein, activating the G protein. The activated G protein binds to adenyl cyclase. Then when GTP is hydrolyzed, it activates the adenyl cyclase. So this is like a process. You have to slowly see word by word, sentence by sentence. Make your own notes. You try to see it. You try to see it. Then you will know. Okay. The next one will be the next part, photoperiodism. Photoperiodism, there's nothing much I will say, but always remember there is two parts. First, you have to identify whether it's a short day plan or long day plan first. Please don't get mistake. Identify the question first. If for a plan they give and you don't know whether it's short day plan and long day plan, then you will take the risk 50-50. This question is very easy because they never mention the plan name. If they mention the plan name and you don't know, then you are taking a 50-50 risk. Yeah. Then all these hormones, please memorize if you try to attempt to do. If you try to attempt to do the hormones, okay. Full name, G protein, and so on. So these are the standard one. They know. If the question, one of your friends asks, one of your friends, sir, for the signal transduction one, is it necessary to write the full name of GCPR, G protein, and so on? It is understood. Like for this one, I will tell my students, if they are weak, they write there G protein couple receptor. If you cannot, you just write there GPCR. Because I don't think so. They will ask you name, let's say this is label X, name X. If the question say name X, so sorry, you have to write the whole name. G protein couple receptor. If they ask you what is X, then you can write there GPCR. What is X? GPCR. If they ask you name X, you have to answer G protein coupled receptor. So sorry. Then for this one, GTP, ATP, cyclic adenosine monophosphate, this one, it is expected of you to know already. So should be no problem. Should be no problem. Okay, should be no problem. Okay, going back to photoperiodism, for this part is actually you have to know this one only. PR, PFR, this one, rapid, this one red light, this is far red light, this is also rapid, then this is slowly in darkness. What does this one do? What does PFR do? What does PR do? PR stimulates which one? Short day plant to flower or long day plant? You have to know all these things. Inhibit or whatever, you have to know. So actually, to flower is actually trigger florigen. 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 So for hormones, auxin and cytokinin, this one you all have to really take note. You have to, this is plant hormone still, nothing much. So I will go to chapter 10. I'll be fast already, I left. For plant hormone, if you don't memorize, it is very difficult for you to do. And until now, the only one that haven't come out is ethylene gas. That's all, ethylene gas. The active and the non-active one. So one of the student asked again, for phytochrome, is it correct if both phytochrome is referred to as active form, depends on the type of plants? Okay, I believe you have read more. So generally, I don't think so. They are asking until in so much detail. So if you read more, yes. If you see, these are the expected answer. I don't say this is the marking scheme. This is the expected answer. So if you look carefully, everything is not in detail, because it's as long as anything that you write is in Campbell and the one marking will can refer from Campbell, then it should be okay. It should be fine. But sometimes I would say that look at the marks. 8 marks, short day plant, long day plant. That means 8 marks, 4 marks, roughly we know 4 marks is for short day, 4 marks for long day. I don't think so is until that detail. Okay, so let me move on to chapter 10. Chapter 10, the first part is the 3 different types of hormones. You have to know very well everything. So let me write it here since I have no. So I know the difficult part. Menstrual cycle. Menstrual cycle, please take note. GnRH, estrogen, progesterone, LH, FSH. There are 5. 5 hormones here. Until now, menstrual cycle haven't come out. So please take note. And early pregnancy, hormones during early pregnancy. What are the late pregnancy? The hormones, early pregnancy. Okay, so early pregnancy, you need to learn how to write. How come? Okay, the hormones for early pregnancy, H, C, G. It's a small H, so estrogen, progesterone. So late pregnancy, they are all together, there are a lot. You should refer to your book. There are a lot. Obviously, estrogen, progesterone, prolactin, then oxytocin, prostaglandin, then HPL. So these are all in late pregnancy. So please take note, when you answer for estrogen and progesterone, they appear every single time. But actually, their function differ at the different time period. So please be careful in exam. If they ask you for menstrual cycle phase, you answer the role of estrogen and progesterone in menstrual cycle. If it's early pregnancy, you discuss about early pregnancy. If it's late, you discuss about late. Then this is parturition. Parturition came out in structure, so no problem. If you ask me, so there's only 2 positive feedback mechanism. It will be in your parturition and ethylene ripening of the bananas. Once it's ripened, it will keep on ripening. It cannot. There's only 2 you all know in your syllabus, positive feedback. So embryonic development in seed and formation of fruit. So embryonic development, you have to mention correctly. So actually, I feel very bad because now today is like too last minute. Usually I will ask for 4 hours for lessons so that I can finish everything. So we have no time for 4 hours. So this one, zygote, basal cell, apical cell, terminal cell, apical cell, then they become what? Then the embryo, suspensor. So embryo, what is all the stage? Globular stage, heart-shaped stage, torpedo-shaped stage. All these things must be memorized. And the suspensor will be crushed by the cotyledon. So please know all these things. This one came out, so I'm not really particular about it. So please be careful with the question, seed germination and embryonic development. Both are different things. Please do not confuse yourself. This is when the question asks seed germination, you answer seed germination. When the answer asks embryonic development, you answer embryonic development. Don't get confused. Then this is gibberellin and seed germination. External factors that influence seed germination. Okay, growth curve. I want you all to particularly pay attention at the growth curve. The one that came out before is the structure question. So if essay, long time already never come out for essay. So if they ask you, if they never ask you with the aid of label diagram, don't go and waste your time and draw all these 3 graphs. Because the previous one that came out for this part, chapter 10, growth curve, the previous one that came out in chapter 10.4, 17 ulangan. So 17 ulangan came out. Then only in 2003, they say with the 8. With the 8, that means everything you draw, label correctly, draw correctly is only 1 mark because you have to describe some more. This one, drawing only 1 mark, drawing and labeling correctly. The other one is 2 marks is for describing. So usually, like this one, repeated after many, many years, 17 ulangan came out, explain. So you draw, you are wasting time, no marks will be given. Explain the 3 types of growth curve. So you need to explain. Anything wrong? So nothing. this part. Okay, this will be on growth curve. You have to know what is the shape: sigmoid curve, bell-shaped curve, declining curve. So look carefully. I know students have no problem with time, the x-axis, the y-axis. This is your height. This is increase in height. This is the percentage increase in height. So these three, you have to know what is the parameter for y and x, then you have no problem. Okay, growth patterns. For human exhibit 2 limited and the other one is allometric. Fish unlimited and isometric. So you have to describe. Then for insects, this is human. I believe after this you can look at, I think you can scroll at the, you can pause in YouTube for tomorrow or whatever, then you can do. What I want you all to do is tonight you rest after my lesson, then tomorrow see whatever you still cannot, then you check again. Now, this is very important. Although might or might not come out, but please take note. From Campbell, produce neurosecretory cell in the brain, produce brain hormone, then release. Juvenile hormone is secreted. Epdyson is secreted. Why? Look at all these things. Explain how hormones inside regulate the ecdysis process. This is the ecdysis process, the name of the definition. So these are the three hormones involved. First one, this is the first essay for your brain hormone. This is the second essay on ecdyson, second paragraph, ecdyson. Third, on juvenile hormone. And this is the one, the whole process. Look carefully. And this one come out. This one is, I believe, last time when the essay come out is 15 marks. So we have to write a lot. Nowadays, it's just 7 marks. For example, 2016, 7 marks only. So you have to write the 3 conditions as well. The 3 conditions. See? High juvenile, low juvenile, intermediate juvenile, low juvenile, and no juvenile. Juvenile hormone, look, low juvenile, and no juvenile hormone. So they are the condition you have to mention. So why I asked this one, you see, the terms such as secrete and produce were wrongly used. So this is how you want to get your A. You have to make sure, how I say, you see, in other words, they just simply use any word to describe hormone production. They are very particular with all the secretion, produce, and secrete, produce, release. These 3 things. So I don't want to dwell in it because it came out and it repeated. So look carefully, 2015 ulangan. Then I thought 2015 ulangan, it won't come out already. So the only one repeated is this part. Okay. Justify or evaluate. Okay, one of the question asked from your friend. Maybe a bit about how do I correctly answer question that asked by using justify or evaluate. I believe this question will not ask about the justify and evaluate. I think is only your school teacher will ask in school to differentiate you all. In STPM, usually explain, describe. Because this one, justify and evaluate, usually is the highest order of thinking skills. So the marking scheme will not be, because there's no correct answer one. As long as you can prove your point, yeah. So I don't think so it will be asked in STPM. Next, the next question is how to calculate growth rate curve. Okay. One of you asked growth rate curve. Since I'm finished this one already, so I can go back. Growth rate curve. Relative growth rate or growth rate curve. So this one, 7 minus 2 will be 5. This is your changes in height. Okay, I think I answered that question. So I will move on. Okay, chapter 11, the very important thing will be this one. The first part will be the first part. The one in your syllabus is only your thermal regulation. That means this question. In how the homeostatic control system in human works in hot environment. If you look at there are so many points because these are all of the old syllabus, old STPM. That's why old STPM, the marking scheme will be a lot. But now you all just need around 7 or 8 marks, so I believe should be okay lah. Then the next question is carbohydrate metabolism. If you look carefully in chapter 11, homeostasis. Homeostasis, important just come out. Definition haven't come out. That's why I asked you all to memorize the word of the definition, but I doubt it will, since you see 2023 or 2023, 19 ulangan, 2022, mammals regulate body temperature during hot, sunny. So the one I'm going to concentrate more will be on your metabolism. This part is still empty, your structure. Then the rest, 23 ulangan, is all very recently only. Very recently, except for Cori cycle. So if I'm referring to this one, did I put it inside? This is the thing. You have to know the whole process. So glycolysis, glucose to pyruvate, so no need to your glycolysis process, I don't want to enter. So these are the 4 processes that happen in a carbohydrate metabolism. So please take note. If you have excess glucose 6-phosphate, it will be stored. This is, okay, excess glycogen. This is insulin. Genolysis, when you don't have sugar in your body. So glucagon will convert glycogen into glucose 6-phosphate, and this is epinephrine and cortisol. Epinephrine is during, or adrenaline, during the stress of threat and extreme starvation. Neogenesis, actually, neogenesis is on, there's 3. 1, triglycerides, proteins, and lactic acid. So it depends on the question. But the key word is always gluconeogenesis only happen when all the glucose in the body is exhausted. You must mention that the glucose in your body is exhausted, so they need glucose from other sources. So actually 1 point is given to them. You must say your body will only take glucose from the fats if there's no more glucose in your body. That's why during fasting, you become slimmer. sometimes you become, this one is only when you break down your fats. So gluconeogenesis only happen when all glucose in your body is exhausted. So they take from triglycerides. Proteins rarely happen. Protein is during, that means, have you heard about the word muscle wasting? It's only during extreme starvation. And if you fasting, I don't think so there's any protein wasting. So you cannot mention about gluconeogenesis from protein. So it depends on situation. So you have to read the situation carefully. Lactic acid is when you are athlete, you're running, after running. So the one I want to concentrate is not this part. I have no time, that's why I have to speed up. The only part will be this one. You have to see about this one. Yeah, the structure. Be careful with all the structure for transamination, deamination, and urea formation. You have to memorize the keto acid and amino acid. How is it? So 20 ulangan, yes, just 20 ulangan just came out a full essay. So are you going to concentrate on it? I'm not sure. So please take note. Conclusion for protein metabolism, there are 4. 1, 2, 3, 4. Transamination, deamination, urea formation, and synthesis of plasma protein. So the next part will be your kidney. Ultrafiltration, selective reabsorption, active secretion. These are the 3 processes. So please take note. A lot of, I saw this year, surprisingly this year, all the questions they ask about this one, glomerulus. So you must know, if this question do come up, you must know why afferent, efferent, how is it pushed, how. One is bigger, one is narrower, but bigger. The lumen is narrower. So what happened? What can be pushed out, forced out? What cannot be forced out? You should know. Then they form the filtrate. So a lot of past year, they are all, I saw a lot of past year on glomerulus, ultrafiltration. So selective reabsorption, yeah, selective reabsorption is here. Then to me, countercurrent multiplier mechanism, if you read it, is actually you explaining, this is your loop of Henle. You have to learn how to explain loop of Henle. Actually, this is the first process that happened. This is the second process that happened. This is the third. Describe the structure. What the structure? This is your NaCl comes out, active transport. Then water comes out. Then you have this concentration increases. Your concentration in your loop of Henle increases. From where to where? This is cortex. Here is your medulla, inner medulla. So then, because the concentration here is high, NaCl move out. This is passive facilitated diffusion. So you have to learn how to know how to write. Write in the correct order, then you have no problem. So this is nothing. Ornithine cycle. So same question. If the question asks, describe the ornithine cycle, will be 5 to 6 marks. Why? The reason is ammonia. Ammonia produced by deamination is toxic, so they have to be converted to urea, which is non-toxic, so to be excreted as urine. So if they never ask you to draw, you don't go and waste your time draw. 6 marks is on the 6 point. That's all. 6 marks is on the 6 point. Is the enzyme compulsory? For this part, I don't one student asked, is the enzyme compulsory? So I don't think so. Certain, how do I say? This one, wait, this part, no need. No lah, it's too much. You have to know that it's too much. So if you cannot remember, then forgo about the enzyme. Important thing, you get the facts right first. Yeah. Then, yes, this one, ADH. Please take note about ADH and your RAS system. RAS system, ADH, please take note. ADH, we talk about osmolarity. Osmolarity. So this essay will tell you the whole answer for this structure, can help you in the whole essay already. This is a past year, so it's a very good question. So we'll tell you everything about this is release, ADH is released to increase. So what happens at this part? If there are more marks, if let's say this question asks, describe briefly how hormone X control the increase in blood osmolarity, 5 marks. If there is more marks, 7, 8 marks, you have to talk how is the aquaporin is formed. How does ADH? I think I have the picture, but it causes the, it is in Campbell, ADH, if there is more marks. Carbamide phosphate. Usually, if student ask me, I will always ask them, refer to Campbell. If you have any doubt with any question, that's why I always refer to Campbell only. So there is nothing wrong. I believe you all have the PDF version already. Okay, the next part. Now, for this one, you have to understand the RAS system very importantly. RAS system, aldosterone. You see, aldosterone. When we talk about this one, we talk about blood volume plus blood pressure, volume and pressure. So, angiotensin 2. First function of angiotensin 2 is to stimulate aldosterone, vasoconstriction of the arteriole. Vasoconstriction so that your blood pressure can increase back. Blood pressure. So important thing is, you see, aldosterone stimulates sodium uptake. Then it causes aquaporin. This one, blood pressure. Please take note. And angiotensin 2, number 3. Thirst. It stimulates thirst. Dahaga untuk air. So these are the 3 things. So eventually your blood volume and blood pressure will all increase. That is your RAS system. Please take note of the RAS system. And ADH, nothing much. Okay, this part, 05 and all the 5. Okay, I will go to immunity. Immunity very fast. If they ask for lymphatic system, so your lymphatic system is this part. Actually, for immunity, you have to memorize the 5 antibody. If you can understand the 5 antibody, then you should have no problem. Then for this part, yes, the one everyone asked me is about antibody should have no problem. The one everyone asked me is about they don't understand. So when pathogen comes, cell-mediated and humoral. If you talk about cell-mediated and humoral, please refer. People ask me, is MHC all this important? Yes, very important. So for example, like you see for this part, what happened? This is the activation of helper T cell. The first part, this part is activation of helper T cell. Pathogen is engulf, and NPM report say they want and digest into antigen. The antigen is presented on the MHC protein. So now this is known as antigen presenting cell. Okay, when they are known as antigen presenting cell, so they start secrete interleukin 1. Okay, interleukin 1. Then the bound virgin helper T cell will secrete interleukin 2. Interleukin 2 got 3 functions. Interleukin 2 got 3 functions. It stimulates the bound helper T cell to undergo clonal expansion, proliferation, and differentiation to form effector helper T cells and memory helper T cells. The next one, it also stimulates your B cell. It also stimulates your cytotoxic T cell. Your B cell to become plasma cell and memory B cell. Cytotoxic T cell to become effector cytotoxic T cell and memory cytotoxic T cell. It is a very clear one from Campbell. Campbell gave a very good one. Yeah. Then once you understand this one, you will have no problem with answering the rejection MHC. Then please take note of all the action. When you talk about humoral, please take note about the action of the antibody. And this is for infectious disease. I have no problem with infectious disease, but the one I want you all to answer, please take note. If the question asks for this is the social prevention, this is the economical, this is the biological. Please learn how to, because they are very precise. The Huraian Sukatan, the curriculum specification, they say you have to describe based on this one. So I'm worried when you write your answer, you write all about this one, but then maximum you can get only 2 marks because they divide it very specifically. So please take note for the question at chapter 13, if they, what are the cause, what are the prevention, you don't give a one-word sentence unless they say that if we divide into 3 but place in the wrong column, will it be, you cannot write in the column. You have to write as essay. Oh, what do you mean by, oh, you write in a try not to, because biological is to remove the mosquito, then social is all those social prevention, economical is cheaper, you feed, you apply mosquito repellent, use mosquito coil. Yeah. So you have to be careful. So that's why this part, I think I'm not going to ask your teacher about it. So what is the definition of infection? You have to know. Infection. What is the meant by term infectious disease? Infection disease. Then if you ask me, everything of it came out already. If you want to see carefully, the one that haven't come out for chapter 13, if I have time, I will discuss with you all. Maybe I will give you a PDF on the cell-mediated and humoral. But for infectious disease, what haven't come out? You look carefully. The one that haven't come out in structure will be dengue. But dengue just come out last year. So if you ask me to read, yes, structure haven't come out. So you must know what is the 4 serotype, then 1, then 2, then 3, then 4. What is hemorrhaging dengue fever? The structure haven't come out. You see, the rest of the structure all came out. Malaria came out twice. TB came out twice. You look at the years. Cholera, yes. Dengue structure, I'm still thinking. Will be this year? Maybe not. But for essay, the one essay that haven't come out will be the transmission. Causes and symptom. Yes, this one, causes and symptom, a lot of students will not get marks because the symptoms they go and write. If they ask for what are the symptoms, fever, full stop. Rashes, full stop. The question say describe. Describe. They never say state. State the symptom. Yeah, if they ask you to state the symptom, you write there, fever. The person is having fever, comma, rashes. This one, acceptable because the question says state. But if they ask you to describe, that means everything has to write a sentence. Like for fever, that means you have to explain biphasic fever. Why is it called biphasic fever? Because you cannot use the term fever come, fever go. Yeah, that is the rashes. What is the rashes like? You cannot rashes full stop. So you have to be careful if they say state. And you have to careful, the rest are all explained. Back to the question that one of the students asked, evaluate and justify. I don't think so STPM will ask until that level. So I think I will end my thing for tonight. How do I? So is everything? Any more questions? Hello? So should I end it or how? Any questions? So will you forward this in the Telegram group? What do you mean by, forward the slides? I will ask YPC to do it, I think. So is it necessary to mention the role of yes, GnRH? There are 5 hormones. For menstrual cycle, there are altogether 5 hormones actually. Gonadotrophin releasing hormone on its role to stimulate, correct? They are altogether 5. I think that's all good. Is it I'm supposed to end it or how?
Oh, I mean, if there's no other questions, I can take
Okay.
over.
Is there any more questions? I think I'll pass back to you because I believe they need to sleep already because tomorrow is their exam.
All right. Okay, then I will take over then. So terima kasih diucapkan kepada Mr. Teo di atas perkongsian yang cukup jelas pada hari ini. Tidak lupa juga ribuan terima kasih kepada pelajar-pelajar dan guru-guru STPM 2024 yang mengikuti program kita pada malam ini. Minta pelajar-pelajar untuk mengisi kehadiran pada hari ini. Klik pada link yang disediakan atau scan pada QR code yang dipaparkan. Untuk makluman para pelajar, seminar yang seterusnya adalah mata pelajaran pengajian perniagaan akan berlangsung pada 14 Ogos bermula jam 8 hingga 10 malam di platform Zoom dan YouTube. Jangan lupa klik link pendaftaran untuk mendaftar. Seterusnya, jangan lupa juga untuk follow media sosial kami di Instagram, Facebook, TikTok dan subscribe channel YouTube YPC International College untuk mengikuti seminar yang seterusnya. Terima kasih dan selamat malam semua. Jumpa lagi pada program yang akan datang. Terima kasih cikgu untuk kelas malam ni.
Thank you.